To understand the structure of DNA, we should first understand its basic components. DNA is made of a series of nucleotides. Each nucleotide is composed of a sugar, nitrogenous base, and phosphate. Nitrogenous bases are what encodes information in DNA. There are 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. Within these 4 bases, there are 2 categories: purines and pyrimidines. The purines are adenine and guanine, and identifiable by a 2-ring structure. The pyrimidines are cytosine and thymine, and are identifiable by a 1-ring structure. These bases pair up with bases from the opposite strand of DNA and are held together by hydrogen bonds. Differences in the amount of hydrogen bonds gives rise to complementary base pairing. Adenine and thymine have 2 hydrogen base bonds, and so paired with each other, and guanine and cytosine have 3 hydrogen bonds, and so they also pair with each other. Nitrogenous bases are also attached to deoxyribose sugar. There are 4 carbons in ribose sugar which play important roles. The 1' carbon is where nitrogenous bases attach. The 2' carbon is how we can distinguish between deoxyribose found in DNA and ribose found in RNA. In DNA, there are 2 hydrogens attached to the 2' carbon, while in RNA, one of these hydrogens is replaced by oxygen with a hydrogen attached. The 3' carbon binds to phosphates from other nucleotides. The 5' carbon is where the phosphate is attached to. The phosphates which originate from 5' carbon and attach to 3' carbons provide a backbone structure for each strand of DNA. When 2 strands come together to form a double helix, they do so in anti-parallel fashion, meaning the 5' to 3' carbon direction will be opposite for each strand. The nitrogenous bases are then stacked flat, parallel to each other and perpendicular to the sugar-phosphate backbone. This base stacking maximizes the hydrophobic interactions in the DNA helix, excluding water and providing stability to the helix. When DNA is twisted together, we see that there are large gaps and smaller gaps in the helix, known as major and minor grooves respectively. It is important to note that in the Watson-Crick model, DNA is right-handed, meaning that when looking at the helix it appears to twist upwards in the right direction. DNA doesn't always stay in the structure of a double helix. It can become denatured and separated into 2 strands, which happens when the hydrogen bonds are broken and the helix is destabilized. However, if the destabilizing forces in DNA are neutralized, then denaturing can be prevented. One way to stabilize DNA would be to put it in a high-concentration salt solution, such as saline sodium citrate buffer. The salt ions would neutralize the phosphate backbone, decreasing the destabilizing forces. With the DNA more stable, the T sub m, or the point when half of the DNA is denatured, would be increased.